Hello and welcome to Assay Genie. Today we are excited to introduce our 3D Organoid Viability Assay, a highly validated assay for your 3D culture research. Why is it essential to test organoid viability? Testing organoid viability provides crucial insights into cell health, growth, and response to treatments, paving the way for breakthroughs in disease modeling and drug discovery. Our assay also plays a crucial role in personalized medicine and optimizing culture conditions. A major challenge facing scientists is the limited availability of sensitive, reliable assays for measuring viability or cytotoxicity in 3D and organoid cultures. And that's where we come in. Assay Genie's 3D organoavailability assay is a highly specific and validated solution designed for use with various organoid types. From neuronal to airway and tumor organoids, this non-lytic low toxicity reagent has been independently validated for 2D and 3D cultures. Our assay is simple to use. Just add, mix, and read. It's fast, delivering results in as little as 1 to 4 hours. Plus, it offers flexibility, allowing additional experiments to be conducted on the same cells. Compared to traditional MTS, MTT, PRESW, or XCTD assays, our water-soluble reagents are more stable, provide a wider linear range, and exhibit higher sensitivity. The assay uses WST8, a compound reduced to an orange formation by mitochondrial dehydrogenases, in the presence of electron coupling reagents. The intensity of the color directly correlates with cell viability, which can be measured at 450 nanometers. In our test with HeLa 2D cultures, the assay demonstrated near-perfect linearity, reflecting its accuracy and reliability. In a case study with apical out airway organoids, or one-step single reagent assay delivered outstanding results, enabling multiple readings over 24 hours. Scientists were able to measure variability simply and effectively. One of our satisfied customers highlighted how our assay outperformed resassurin-based methods for airway organoid viability. Thank you for watching. If you want to learn more or you have any questions, please visit our website or send us an email to info@assaygenie.com.